Okay, so this next section talks about the internal cellular structures. Learning objectives are listed here. Part 1 talks about the basic cellular structures and organelles. So in the last section, I started to talk about the cell membrane, and I was talking about how many different types of proteins are found in the cell membrane. So although we draw a cell membrane typically as just being a circle, there's a lot of stuff going on within that circle. Now, cells need to be able to form tissues, and in order to connect together to form those tissues, we have different types of cellular connections. These are those adhesion proteins that I talked about in last week's lecture. The three major types of adhesion proteins and cell connections include desmosomes, gap junctions, and tight junctions. Desmosomes are button-like connections. So they anchor cells together, kind of like a button would hold a shirt together. It's a loose connection. Molecules can definitely move between the two different cells, but it's just holding them together. Many of your tissues are held together with desmosomes. The next one is gap junctions. So with gap junctions, you have a tunnel or a channel that physically connects the cytoplasms of adjacent cells. So with a gap junction, whatever's in one cell can float through into the other cell. Gap junctions are found in between the cells of the heart. So if we have electrical activity happening in one heart cell, we want it to get to the neighboring heart cell very quickly so that we can have an overall contraction of the heart muscle tissue. So we have gap junctions there to allow for the transfer of information from one cell to the next very fast. Then we have tight junctions. With tight junctions, the cells are kind of sewn together. So when they're sewn together in this manner, nothing can pass between the two cells. These are numerous in the blood-brain barrier because we don't want just any old substance being able to pass from the blood into the brain. So we have tight junctions there, which prevent the movement of substances between cells. Now, sticking with the outside surface of the cell, although cells need to be able to stick together, cells also need to be able to interact with the environment a little bit. And these structures are going to be found on the surface of the cell, and they all do something a little bit different. We'll start with microvilli. Microvilli are extensions of the cell membrane which increase the surface area for the absorption of nutrients. So again, microvilli are extensions of the cell membrane which increase the surface area for the absorption of nutrients. So if we have a typical cell, let's just draw it as a square. And let's say that these are cells of the digestive tract. So we need to absorb nutrients from the GI lumen into the bloodstream through this cell. If we just had squares in here, we would have one surface to absorb nutrients through. But what microvilli are instead of having just regular old squares, they're actually extensions of the cell membrane. So now when we absorb nutrients, we have all of this surface area to absorb the nutrients on. We can absorb it in every single side here so that lots of nutrients can make their way into the center of that cell. So instead of one surface, we have now multiple surfaces. So that's what microvilli are, and here they are pictured up here, electron micrograph picture. There's a bunch of these that extend out from the surface of a cell, and they're going to increase the surface area for the absorption of nutrients, and these are predominantly found in the digestive tract. Cilia. What cilia are different from microvilli. So here microvilli you can see are actually extensions of the cytoplasm. What cilia are just hair-like projections that stick up off the surface. They're like little fringes on a rug, if you want to think of them that way, but these are going to constantly be swirling and moving, and they help brush substances across the surface of a cell. Cilia are going to be numerous in the respiratory tract. When you breathe in air, you want to get all that nasty debris that we've breathed in out of the lungs and out of the respiratory tract. So you have cilia here, and the cilia are going to move and brush substances across their surface to help move debris out of the lungs. You can also find cilia in the ovarian tubes, the uterine tubes, also known as fallopian tubes. They're going to help brush the egg down into the uterus, either so that the implantation event can happen after fertilization, or to eliminate the egg from the body if there is no pregnancy. So we have these hair-like projections that are going to brush substances across their surface. That's what cilia are. Now flagellum is talking about just one of them. Flagella is talking about multiple. The only cell in the human body that has a flagella is the sperm cell. A flagellum is a tail-like structure, and that's going to help aid in the movement of the sperm cell. So the sperm have a really long journey to swim up into the uterus and into the fallopian tube in order to find an egg to fertilize. So these tail-like projections allow them to swim. And that's the function of the flagella in the human body. The only cell that would have it is the sperm cell. In microbiology, we have a whole bunch of other aspects associated with the flagella, but for this class, it's just a tail-like structure that allows for movement of the sperm cell. The cytoskeleton. So just like your body has a skeleton that lays the framework for what all of your organs sit on and how you move and the levers that make your body move, your cells have an internal framework. This internal framework consists of microfilaments, intermediate filaments, and microtubules. Intermediate filaments are the most like your skeleton. So they provide structural support and cell shape. So they're pretty rigid, and they're going to form the shape of the cell. They're kind of like an internal scaffolding that keeps the cell in a particular shape. Microfilaments are the thinnest, and these are going to aid in movement. So if you ever want to have a muscle contraction, it's the microfilaments that are moving past each other in order to contract that muscle. Microfilaments are also going to form the basis of cilia and flagella. Then you have microtubules. Microtubules are large tubes. And you can see how these subunits come together and they're going to form these tube-like structures. Microtubules are responsible for moving substances around in a cell. So if you need to move an organelle from one side of the cell to the next, it's a microtubule that's going to push it or pull it. The microtubule is also going to play a major role in the division of the cell, which we'll talk about at the end of this lecture series. Here you can see microtubules forming up the centrioles, which are found in the centrosome of the cell. So centrosome is simply the cell center. It's an area. It's not necessarily a structure, although there is a structure inside of it that kind of defines that area, but it's the center of the cell. And when we say cell center, we're not talking about the physical middle of the cell. We're talking about the point from which the cell is going to start to divide. If this cell wants to make a copy of itself, it's going to grow really big and split in half. Now this is that half mark. This is that deviating point which will eventually separate and migrate toward opposite ends of the cell and allow for the division of this cell. Centrioles are bar-like structures that form the basis of our cell division machinery. So centrosome is just an area, while the centrioles are bar-like structures made up of microtubules, and they're going to aid in cell division. A lysosome. I like to call the lysosome the mini stomachs of the cell. Lysosomes contain digestive enzymes. So if your white blood cell eats a bacterial cell because you were sick with some type of bacterial infection, it has to be able to break it down somehow. Well, that's where the lysosome comes into play. A lysosome contains digestive enzymes which allow for a cell to be broken down, or a cell to break down structures that are brought inside. So whether it's eating a bacterial cell or destroying a dead or non-functional organelle inside the cell, lysosomes digest. They're going to break down substances. Peroxisomes play a major role in detoxifying the cell. They mediate oxidation reduction reactions, or redox reactions. Now, if you've taken a chemistry class previously, you're familiar with what redox reactions are. But there's all of these toxic oxygen compounds. And when these accumulate in the cell, what they want to do is snatch electrons from atoms. Now, if you have an electron snatcher in the cell, that's very dangerous. It can cause a lot of damage. So we have antioxidants, and antioxidants are going to feed these electron snatchers electrons so that they don't have to steal electrons from your own organelles and they don't cause damage. So antioxidants are definitely good to consume in the diet. They're stored inside of these peroxisomes, and peroxisomes are going to help neutralize these electron snatchers that you'd find inside of the cell. These electron snatchers are generated every time that we produce energy through metabolism. So they're constantly being made. So we need to neutralize them before they cause an issue in the cell. As a person ages, these peroxisomes decrease in number, and mitochondria start to dysfunction, and they start to produce a lot more of these free oxygen radical compounds, and that increases the process of aging. So the more you consume antioxidants, the more you're going to kind of keep them at bay, and the slower you're going to age. Proteasome. Proteasome destroys unwanted or misfolded proteins. If you create a protein, and we'll talk a little bit about the processes involved in creating a protein, but if you make a protein and it's the wrong type of protein, or it's misfolded, or it's not functional, you don't want that hanging out in the cell. That's cellular debris, and it has to be handled in a certain way. We don't want lysosomes to break down unwanted proteins, because lysosomes completely digest and destroy whatever they're coming in contact with. While a proteasome, if a proteasome digests a protein, it'll break the protein down into amino acids, and those can be recycled in the cell. So proteasomes degrade unwanted or misfolded proteins, and then that allows for the recycling of these amino acids. The mitochondrion. A lot of times you'll hear textbooks and other resources call the mitochondrion the energy center of the cell. So we're going to take a minute to talk about what is energy. If you think about what is energy, energy is a movement, or it's the ability to cause a movement. When the body ceases to move, that's death. There's no more movement, that's what we call death. So energy is the ability to cause movement, or it is the movement itself. So we have potential energy, again, that's the ability to cause something to happen. And we have kinetic energy, so that's the energy of actually moving something. So how do we get something to move in a cell? Well, we have this molecule called ATP. ATP stands for adenosine triphosphate. This adenosine group actually comes from the base adenine. So this is derived from the bases that make up our DNA. We also have other types of energy. There's GTP, UTP, and those are used very rarely inside of the cell and for very specific functions. So if you were to take an advanced metabolism class, you'd be hearing about the different DNA bases and how they make different triphosphates and what those are specifically used for. But for general A&P, you just need to know about adenosine triphosphate. This is the most common compound that we use for energy. Now we have this adenosine group again that's a derivative of the base adenine, and we have triphosphate. Tri means 3, so you can see 1, 2, 3. The way that this thing produces energy is by breaking off this last phosphate. When we break off this last phosphate, it causes a spark, which can cause a movement. Kind of like if you know anything about engines, when you throw gasoline on a piston, it causes a spark, so it ignites, and it causes that piston to move. So this causes a movement. So we go from adenosine triphosphate, if we break off the phosphate, 3 minus 1 gives us 2, gives us adenosine diphosphate. So ATP has 3 phosphate. When we hydrolyze it, we break it down into ADP, which has 2 phosphates, plus the free inorganic phosphate. Now we want to make energy again. So we want to put these back together. So we caused our spark, now we have to regenerate ATP. When we bring those back together to make ATP, that's the process of cellular respiration. This process is going to be discussed once we get to the respiratory system because it involves oxygen and carbon dioxide, and it makes sense for me to discuss it once we get to respiration. But how does this affect what the mitochondrion is doing? So this is a picture of a mitochondrion, and although we can produce energy outside of the mitochondrion, inside of the mitochondrion, this is where a majority of the energy is produced. There's this thing known as an electron transport chain that you'd find right in the cell, the inner membrane of this mitochondrion. It's inside of here that you will have this process going on, where we will generate ATP. And we make the most ATP from this electron transport chain. So again, although I'm going to discuss it more in the future, right now what you need to know is mitochondria is responsible for producing the most ATP in the cell. Without a mitochondrion, you wouldn't be able to survive. So you need to produce a lot of energy as a multicellular organism, as a eukaryotic organism. We have mitochondria in order to produce that energy.